What is ATP synthase?
ATP synthase is an important enzyme that creates the energy storage molecule adenosine triphosphate. ATP is the most commonly used energy currency of cells for most organisms. It is formed from adenosine diphosphate and inorganic phosphate. Energy is provided by the hydrogen ions moving down an electrochemical gradient from the intermembrane space into the matrix of the mitochondria. Any significant mutation in this enzyme could cause detrimental effects on the amount of energy being produced in the organism's body, so alternative pathways such as the Krebs cycle will be used for the body's source of energy. The F-zero subunit is a cylindrical structure capable of rotation when driven by protons through a revolving central stalk inside the F1. Rotation is derived from the C10 ring inside the F-zero subunit, which captures the protons coming from the inner membrane space and translocates them to the matrix. About 10 protons give one complete revolution of the C10 ring.
The F1 subunit of ATP synthase contains the sites where ATP is synthesized. The proton flow from F0 spins the gamma subunit, which induces a conformational change across the beta sites in F1. These conformational changes cause ADP to bind, ATP to form, and ATP to release. F1 is held in place by the peripheral stalk, preventing it from rotating randomly. The stator ensures that the F1 catalytic head does not rotate with the motion of the central stalk in F0 and must be strong enough to resist the tension that builds up in the central stalk between each 120-degree rotation of the F1. Subunits E and G produce a 100-degree membrane curvature in the dimer that allows the F subunits to interact and bind. The formation of dimers allows close packing of ATP synthase in the mitochondria by forming cristae. This close packing ensures that mitochondria can produce enough energy to power the cell. ATP deficiency is commonly associated with a genetic mutation. Mutations can be either in genomic DNA or mitochondrial DNA. ATP synthase deficiency is accompanied by symptoms including tissue dysmorphism, cardiomyopathy, metabolic crises, and 3-methylgluconic aciduria or 3-MGA. Patients have been observed to have varying degrees of metabolic crises. In some cases, lack of ATP synthase leads to early death, while in other cases, patients survive for longer periods of time with development issues. The problem has been addressed by adjusting the diet of the patients. Sugars are almost completely eliminated from the diet to avoid the formation of lactic acid as pyruvate builds up in the body. The ingestion of fats and proteins is increased together with a supply of different vitamins that are essential to help the metabolism of the patient. The name anaplerotic diet refers to the fact that this diet intends to supply materials for side reactions of the metabolism of the patient. These side reactions are not the main source of energy for healthy patients, but are important to ATP deficient patients.